Good day, everyone. Today we'll explore the important equipment used in bacteriology, many of which you've already encountered in the laboratory. After that, we'll go deeper into microbiology automation systems such as API, Vitec, BactAlert, BD systems, Microscan, MaldiToF, and more. Automation is now central in clinical microbiology because it helps us work faster, safer, and more standardized while still keeping our critically thinking skills sharp. So let's begin by revisiting the basic equipment used in a bacteriology laboratory. These are foundational tools that automation is built upon. So think of this lesson as levels. So level 1, manual tools. Level 2, semi-automated tests such as your API. Level 3, fully automated analyzers such as your Vitec, Phoenix, Microscan. Level 4, rapid identification such as your MaldiToF. And level 5, automated blood culture. You have your BacTech or your BactAlert. So this helps you mentally organize everything. So shown here in the picture are the different equipment used in bacteriology. So you have the microscope, you have your hot plate, and as well as the disposable petri dish and the disposable wire loop. Next, aside from those mentioned, we also have the following. So you have your water bath, microscope, the biosafety cabinet, Bunsen burner, etc. Now let's proceed to your analytical balance. It is a sensitive weighing device used for measuring extremely small masses down to sub-milligrams level. Notice the transparent enclosure, so that prevents dust, air currents, or your breath from affecting the accuracy. In bacteriology, we don't weigh items daily, but balances are essential in preparing accurate culture media, reagents, or buffers. So please take note in order for you to remember analytical balance. Analytical meaning analyze tiny amounts. So the glass enclosure is your hint. So because it keeps tiny weights stable. Next, we have your autoclave. It is the heart of sterilization. It uses steam and pressure and time to kill even bacterial spores. So the gold standard parameters are 121 degrees Celsius, 15 per square inch, or the 15 psi, and as well as 30 minutes if for decontamination. But if for sterilization, it is 15 minutes. So everything must be exposed to saturated steam to be properly sterilized, and this includes media, glassware, instruments, and even biohazard waste. And please don't forget also the biological indicator of your autoclave. It's your Geobacillus or Bacillus stearothermophilus. Next, we have the Bunsen burner. It produces a direct open flame used for sterilizing loops and creating a sterile field. But because of biosafety concerns, many laboratories now prefer the bacterial incinerators, the one on the upper portion of this presentation, which use infrared heat at 850 degrees Celsius, so achieving sterilization in 5 to 8 seconds without aerosols. Next, we have the centrifuge. It separates components of liquid based on density by spinning them rapidly. In microbiology, we use centrifuges to concentrate bacteria, sediment, yeast, or prepare specimens for culture and staining. Next, we have your colony counter. It is used to count the number of colony forming units or CFUs on plates. This is essential for urine cultures, quantitative cultures, and determining bacterial load. Next, we have your hot plate. It is a tabletop heating system for gentle heating. It is usually ideal for warming media or reagents without an open flame. Next, we have the magnetic stirrer. It mixes solutions using a magnetic bar that spins inside the container. So it's quiet, it avoids contamination, and maintains consistent mixing. Next, we have the hot air oven. So it sterilizes items using dry heat, which is great for glassware, powders, and then metal instruments. So it destroys microorganisms and spores, but requires longer times than the steam. And please don't forget the biological indicator of your autoclave. I mean the oven. It's your Bacillus subtilis variant Niger. Next, we have the incubator. Incubators provide controlled temperature, humidity, and atmospheric conditions. So they allow microorganisms to grow at optimal conditions, usually 35 to 37 degrees Celsius for clinical bacteria. So think of your incubator as a bacterial baby crib. Next, we have your biosafety cabinet. It protects you, the sample, and the environment. It uses HEPA filter or the high efficiency particulate air filters to provide sterile airflow and may include UV light for decontamination. So this is essential for handling pathogenic organisms. So think of the biosafety cabinet as easy as PPE. So personnel, the product, and then the environment. So it is a three-way protection. So we have three types of biosafety cabinets. We have class 1, class 2, and class 3. For class 1 biosafety cabinets, the protection, it protects the personnel and as well as the product. But I'm sorry, it protects the personnel and the environment. It has no product protection. Okay, so please remember that. Again, only the personnel and the environment is protected by Class 1 BSC. The airflow, it's inward only, just like a fume hood. So the HEPA filter is on exhaust, and then air is not HEPA filtered before heating the sample. Typical usage of class 1 BSC, it's low to moderate risk organisms. So you can also have, you can changing stations, and then it is not suitable for sterile work because the product is not protected. So please take note of this key idea for class 1 BSC. It protects you and not the sample. Now let's proceed to the class 2 BSC. For the protection, it protects the personnel, the product, and as well as the environment. So everything. So the airflow is inward, and then plus it has HEPA filtered laminar downflow. So both intake or the downflow and the exhaust air are HEPA filtered. So typical use is usually for clinical microbiology laboratories, handling pathogenic organisms such as biosafety level 2 and some biosafety level 3 organisms. Most bacteriology work is done here. So again, please take note of this key idea that BSc Class 2 protects everyone and everything. So this is the standard BSc for laboratories. And for the Class 3 BSc, the protection is the same, everything. So it protects the personnel, the product, and the environment. So Class 3 BSc provides the highest level of protection. So the airflow is completely enclosed. It has a gas-tight cabinet. So the work is done through glove ports or the so-called glove box, as you can see on the picture. The air enters through HEPA filter. It leaves through the two HEPA filters. So this is often under a negative pressure. So typical use of your class 3 BSc, it is usually for biosafety level 3 and biosafety level 4 pathogens. So which are very dangerous organisms, no, such as your Francisella, Brucella, and as well as your viruses. So the key idea for BSc class 3, please take note that it provides the maximum protection and it is human proof, or it's just like a pathogen prison. Next, we have your deep freezer. It stores samples long term, including bacterial stocks, sera, and reagents. So this keeps material stable for extended periods. So I have just a quick trick for all of you. So when we talk about deep freeze, it's equivalent to deep time or long term storage. Next, we have the microscope. It's your window to the microbial world. So it provides magnification to visualize bacteria, yeast, and other microscopic structures essential for gram stain interpretation. So, so your microscope is basically your best friend inside the laboratory. Now let's proceed to the automation in the microbiology lab. So we have actually different companies or manufacturers uh, providing machines or fully automated equipment in the laboratory. So first we have your Biomeru. Biomeru, we have your API. So API or Analytical Profile Index, it's a miniatured biochemical identification system. So as you can see on the picture, each strip contains dehydrated media for multiple biochemical tests. So you inoculate each copule with a bacterial suspension and incubate. After incubation, color changes indicate positive or negative reactions. You then generate a profile number that is matched to a database. So please take note of this important key idea that your API is for identification only and not for AST. So again, your API, it tests many biochemical reactions at the same time. So the organism changes the colors in the wells, and then you are going to convert these into a profile number, and then the number tells you the species. Okay, so think of it as a like this, color changes, and then which results into code, and then that code will result into an identification up to the species level. So your API can identify gram-negative rods, yeast, staphylococci, strep, anaerobic bacteria, and other bacterial groups. Next, we have your Vitec 2. It is a fully automated system for identification and as well as antibiotic susceptibility testing. So it uses fluorogenic methodology, which is used for ID, or it detects fluorescence, and the second one is the turbidimetric method for AST, or it monitors turbidity or growth. So results can be ready in 5 to 8 hours. So for the principle, it shines fluorescent light to detect metabolic reactions for identification, and it measures turbidity or cloudiness for AST. Next, here's the procedure on how you are going to prepare a suspension and as well as how you are going to load your sample into the Vitec 2 compact equipment. Next, we have your Backed Alert 3D. So this is an automated blood culture system. It detects growth based on carbon dioxide production. When bacteria in the bottle produce carbon dioxide, the color sensor at the bottom changes from green to yellow. So the machine reads this change through reflected light. So please take note, since this is Backed Alert, so alert, which means yellow alert, no, when there is a carbon dioxide present. So this is just a personal showing. She is going to remove a positive blood culture bottle because it is now yellow. Next, for the principle, I just make it easy for everyone to understand. So there is a bacteria in blood culture bottles. They will produce carbon dioxide, and then the bottom of the bottle has a sensor that changes color when carbon dioxide increases. So the machine reads this color change using light. So just to make it simpler, so more carbon dioxide, the bottle will turn yellow, and the machine will flag it as positive. Next, we have your WASP or the walkaway specimen processor. It automates specimen processing, so it streaks the plates, no, it prepares gram stains and standardizes inoculation. You load the specimen, then you walk away, and then the system handles the routine tasks. So as what I have mentioned earlier, that your WASP, it automatically streaks the specimens, prepares slides, and inoculate plates. So you are going to load the sample, and then the machine does the routine processing for you. So let's take it this way. You're going to load it and then leave it. The next company we have your BD. So the first product that we are going to discuss is about the BD Phoenix M50. So it is a broth microdilution system for identification and MIC based AST. So the principle, it's a redox indicator which detects metabolic activity by color change. So it can work with BD Phoenix app or BD Phoenix AP for automated inoculation preparation. So again, for the principle, let me just make it simpler for everyone. So it uses a color changing redox indicator to detect bacterial growth on broth microdilution wells. So if there is color change, and it is equivalent to organism activity. So again, to make it simpler, if there is color change, then the growth is detected. So this is how you are going to perform it. So this represents the BD Phoenix workflow. Now we have another fully automated blood culture system under BD. So when bacteria grow, they produce carbon dioxide. The carbon dioxide will react with a dye sensor, changing fluorescence, which is measured by a photodetector. So the principle is like similar from the backed alert. So again, I will make this easy for you to understand for the principle of your BD bactec. So the bacteria produce carbon dioxide, and then the carbon dioxide reacts with a dye, and then the dye becomes fluorescent, and then the machine will measure this fluorescence. Okay, it will now be flagged as a positive blood culture bottle. So other products under BD, we have your isolator pack plated media, the prepared plated media, and as well as the prepared tube media. So next, we have the manufacturer, the company Beckman Coulter. So the first one that we are going to discuss is your microscan walkaway. So the microscan performs identification as well as AST using colorimetric optics. So it's a color wheel plus the lamp plus the photosensor. So it gives initial reads at 2.5 hours and then final reads anywhere from 4.5 to 18 hours depending on organism growth. So imagine that is really, really, really fast. So it reads biochemical and growth reactions using a colorimetric optical system, a color wheel plus the light plus the photo sensor. So the color determines the identification and the AST of the organism. So think of it this way. So microscan, so meaning micro colors are scanned. Next, we have your MALDI-TOF. MALDI-TOF, M for matrix, A for ablation, L for laser, D for desorption, I for ionization. TOF, TOF, it's the time of flight. So meaning MALDI-TOF is the matrix ablation, laser, desorption, ionization, time of flight. So MALDI-TOF, it's one of the fastest microbial ID tools today. So it's widely used for identifying bacteria, yeast, and even detecting food fraud. So think of laser shoots, and then it will make your proteins fly, and then the computer will identify it. So the sample of your MALDI is uniformly mixed in a large quantity of matrix, and then the matrix absorbs the ultraviolet light. Usually it has a wavelength of 337 to 355 nanometers, and it converts it to heat energy. So a small part of the matrix down to 100 nanometers from the top outer surface of the analyte, it heats rapidly in several nanoseconds, and it is vaporized together with the sample. So for the principle, again, I will make it simpler for everyone. So a laser hits a matrix embedded sample, and then the molecules desorb and ionize, and then their mass or charge ratio is measured by the time of flight. So the protein fingerprint identifies the organism. Again, a laser blasts the sample, and then the proteins fly into the detector, and then the time they take to fly tells you their mass. So these protein patterns identify the organism. So this picture also shows you on how you are going to process the sample using your Malditov. So please take note, everyone, that all these machines give fast results, but remember that no machine can accelerate the growth of microorganisms, but they can only detect growth faster or analyze it better. And that wraps up our discussion. So these machines help us work efficiently, but your knowledge of manual microbiology keeps everything accurate and meaningful. Automation assists us, but it cannot replace a well trained medical laboratory scientist. And that would be all, everyone. Thank you so much for listening.